All right, good morning. Well, it's my pleasure to introduce Ryan Long. Ryan is a graduate student in the biology program, and he was also an undergrad at JSU in the cell and molecular concentration. And he's interested in genetics and evolution, and he also has an interest in learning Japanese language. He spent a semester abroad at the, is it Kyodai? Kansai, okay, in Osaka. And so a fitting project for him, we thought, was to look at a taxonomic and evolutionary question surrounding a group of bamboos that are native to Japan and sister to bamboos here in the States. So Ryan Long.
Hello, Ryan. So my talk is about the molecular evidence of cryptic hybridization in a group called the Japanese Nezasa bamboos. Right, so first of all, who is Pleioblastus and what is section Nezasa? And so we're all on the same page. You know, taxonomic ranking is kingdom, phylum, class, order, family, genus, species. And then between genus and species, you have section, which is just a little more specific than genus, but broad enough to include several species. And so genus Pleioblastus was first recognized in 1925 by a Japanese botanist named Kai. And then it had about 100 species and 100 plus subspecies. And then Suzuki came along and he reduced it to about 21 species in 1978. And so within this group, Pleioblastus, we have a section, Nezasa, with about 9 contentious species, mostly found in cultivation. And so this kind of raises the question, like, what happened to those 100 species? How was it reduced to 21? What happened there? Like, did those species stop existing, or did we just, begin to see them differently? And so that raises the question, like, what even is a species? So cat, dog, different species, that's kind of easy. But what about all the diversity in domestic dogs? They're pretty different. And what about in the off chance you catch your dog humping your cat? It happens sometimes. it's biology. So these organisms, they don't recognize the boxes we draw around them. Reality just is, and we impose our classification upon it. And so that's where like species concepts come into play. It's like a common one is the biological species concept, which states that if you have 2 interbreeding individuals and they produce an offspring that is fertile, they're a species. There are more, but it gets increasingly complex, in some cases, when what is a species? And so this kind of raises 2 schools of thought in classification and taxonomy. You have lumpers and splitters. Splitter, on one hand, they will look at any distinctive feature and they'll make a classification about that. On the other, you'll have clumpers who they take a more gestalt view and they prioritize the similarities rather than the differences, making one group. And so for the state of bamboo taxonomy in America, we've got 3 main groups, and it reflects more of the clumper approach. We've got Tecta, Appalachiana, and Gigantea. And they hybridize, and to make it more complicated, like plants and grasses and bamboo in particular, they hybridize much more readily than animals. And they do it between species and also between genuses or genera. And then, so Japan, you have significantly more species and you have a lot more hybridization going on. It makes it really complex. And they also take more of that splitter approach to taxonomy. And then China is even more complex. So what's happening in the US is kind of like a microcosm for Japan. In our work with the Japanese bamboo, you can think of it as like a stepping stone for the ungodly complexity of China. Right. So here comes my thesis, and so it didn't exist in a vacuum. It relies on previous work. So over here you've got a little character matrix. You've got species over here, and then for characters you could say like glabrous or hirsute, smooth or hairy leaves. And then you can track them, the presence of these with 1s and 0s. And so with that you can infer relationships. And so Triplett and Clark in 2020, they sought to examine this morphological relationship. So it showed that Nezasa kind of falls out into some groups. But the genetics, they didn't really support that. And the reason why is because these hybrids, they kind of gum up the works in the programs, and it makes it really messy. So these established relationships that we know exist aren't showing up because these hybrids get in the way. So everything in red is kind of like a suspected hybrid. And so enter my thesis. And the objective is to use DNA evidence to test this hypothesis if hybridization is occurring in this group, and to see if some of these currently named species are in fact cryptic hybrids between some other species in this group. All right, so now That's what we wanted to do. Here's how we did it. So sample collection, Dr. Triplett went to Japan. He collected 6 samples and brought them back to the US. All right, so this is supposed to be a video, probably won't play, but this is preparing samples in the lab. You weigh them out, you label them, and you grind them up in a mortar and pestle with some liquid nitrogen, and then you do centrifuge to get this DNA-rich solution. Right, then you do PCR, so you got a little bit of DNA, and then we make a lot of it, so we can work with that a little more better. All right, again, another video that can work. And then AFLP, so this is what we did next. So this is amplified fragment length polymorphisms. So you get this total genomic sequence, you chop it up with restriction enzymes into these little bits, and then with these little bits, you can put them into a gel. And there's like these microscopic holes, and when you run a current through that, these charged DNA fragments will move at different rates. And so they'll clump up, if you chop them up right, into these distinct units. They're bands, and these represent alleles, or different forms of a gene. And so with these different alleles, you can make a giant tree based on who's related to who. Right, and so this is another program, this is called Gene Marker. This is a way to look at those allele bands across individuals. And so each peak represents an allele. And so then you get data output, you get a giant matrix of ones and zeros, presence or absence of these alleles. And so we went through this and we made sure all these bins are accurately reflecting an allele site. And then we also went through and we made sure that these peaks were in fact reading as presence or absence. And so that was a lot of work, a lot of hours of tedious grind. Then we did some analyses. So we did some phylogenetic analyses using POW, which is a computational phylogenetic program that makes trees based off of like parsimony, likelihood, Bayesian inference, all those things. And we also used another way to visualize those trees. It's called split tree. We'll see that in a second. And then we did genotype analysis using a population genetic program called Structure. And we'll also see that in a second. So, good results. So this is a split tree. So this is an unrooted tree. And then so this is all in the genus Pleioblastus. This is section Nezasa. And then this is section Pleioblastus within genus Pleioblastus. And so this is kind of what we want to see. You got like these distinct entities coming out in these clusters. And so this is strong support for the presence of a species. And so this is, as you can see, a bit of a mess. And so if you look at it with just Nezasa, still a mess, but you can see it's kind of falling out into 1 and 2 groups. So Nezasa 1, Nezasa 2. Right? And so this is structure. So this is that program I mentioned earlier. And so this is just another way of looking at that same AFLP data and assigns individuals to specific ancestral populations. So, like, for example, this individual here in yellow is like 99% of this ancestral population, this yellow thing. This color is just arbitrary. And so what this shows us, this is Pleioblastus, this is Nezasa 1, and then this is Nezasa 2. So it's showing us 3 main groups. And then we told the program, hey, do what you do for assuming that there is 2 ancestral populations all the way up to 20. And so at every step of the way, it kept showing us that there is 1, 2, 3 groups. So that's strong support for only 2 sections, or two groupings within the section of Nezasa. And so this leads us to some conclusions. Either 1, there's only 1 highly polymorphic species within Nezasa, or 2, you've got so many polymorphic species and absolutely no resolution in sight, or 3, the one that we suspect is the case. And there are 2 highly polymorphic groups within Nezasa with hybrids between the 2. Right? So our objective was to look at this DNA evidence. Is there hybridization in this group? Where are the cryptic hybrids? And our conclusion, is there support for the current taxonomy? The answer is no. And then is there support for leaky boundaries? Yes. And then most likely we have 2 entities with ongoing lineage sorting between the 2. All right, so some acknowledgements. Thank my advisor, Dr. Triplett, fellow researcher, Morgan Brown, and then my thesis committee, Dr. Turner, Dr. Murdoch, and the JSU biology. So, any questions?
I just have a statement. That was a good presentation.
Thank you.
I like the humor. And it makes me totally look at my houseplants differently.
Where is this stand of bamboo?
This is a picture from Dr. Triplett's fieldwork in Japan. And so this is what it looks like on roadside. It's just like this giant clumpy mass. Yes.
Has there always been a thing called section that's between genus and species?
I'm not sure, but it's like it's common in botany. So there's like subfamily, suborder. There's just like you can keep going.
Yeah.
Yeah.
Are you a lumper or a splitter?
I think I follow the evidence, and whichever one is most
Oh.
appropriate.
Great answer, too.
Yeah, very nice.
Right. Any other questions?
All right.
Thank you.